My work and my projects have been centered around the question of development, which is one of the big unanswered questions in biology. How do you go from a single cell into a functioning organism? A lot of my research has been centered around building tools to help us solve that question. My project here was a microheater system. We have these fish lines that we can activate gene expression in by raising the temperature, but development is very much a spatial and temporally regulated process. There's a lot of patterns and spatial dependence of how gene expression is involved in coordinating the movement and differentiation of cells. So usually when we use these temperature-sensitive fish lines, we can get temporal control, but we cannot get spatial control. So my project was making a microheater system that's small enough to heat up a small patch of the fish so that you can get a bit of spatial control on these established fish lines. The ideal tool would be being able to turn on exactly whatever gene you want in whatever location at whatever time. The closer we get to that ideal tool, the more questions we'll be able to answer regarding animal development, and the more we'll be able to tease apart these hidden programs and patterns of gene regulation that are responsible for driving this sort of beautiful process of animal development. Cells are able to build things much better than we were able to build things. Just look at a skeleton, it's so complicated, and yet it's just made from very readily available materials. If you're able to figure out the language behind how cells are able to coordinate and arrange themselves into new organisms, it would be really awesome to be able to decipher that and learn to use those tools to build stuff for humans. I had some research experience coming into Stony Brook, and I knew that this was something that I wanted to do. So during my undergraduate freshman year, I looked through the biochemistry and cell biology department website, and I found some faculty members that I was interested in working with. So I sent out emails to them, and that's how I got started. I think the most common thing is to show up to office hours and talk to your professor. If you like their teaching style, you think they might be a good mentor, take a look at their research. If it interests you, then I would talk to them. When I first joined the lab, I had a bunch of ideas, but I really didn't know what I was doing. So it's kind of scary. You don't know whether your ideas are valid. You don't know whether you're going in the right direction. I think the most helpful thing for me at that point was just having the space to sort of explore and having Ben willing to give me advice on the different ideas I came up with. He just gave me a lot of space to grow and develop as a scientist, and that was really helpful. Stony Brook has treated me really well. The availability of undergraduate research is fantastic. Through the flexibility of the biomedical engineering major, I was able to build a really helpful foundation of knowledge and intuition for the research problems that I'm interested in. I was able to not only draw from knowledge and resources in the biochemistry and biology department, but also from the physics and chemistry department. And I think having these different perspectives from these disciplines shines new light in the field that I'm interested in, which is developmental biology. Sometimes trying to get involved with things can feel intimidating because you feel like people, oh, they're so established. The opportunities are definitely out there. Try to pay attention to the things that you have going for you. Try to pay attention to the things that you enjoy. If you get lucky enough to get to a position where it feels self-propelling, you should try to grab hold of it.